Welcome, students. Today, we will dive into the fascinating world of gluconeogenesis. Gluconeogenesis is a crucial metabolic pathway that occurs primarily in the liver and kidneys. It allows organisms, including humans, to synthesize glucose from non-carbohydrate precursors, such as amino acids, lactate, and glycerol. In this lecture, we will explore the intricate regulation, molecular mechanisms, and clinical implications of gluconeogenesis. So, let's embark on this journey together. Gluconeogenesis is a tightly regulated process, governed by several key enzymes. The rate-limiting step is catalyzed by the enzyme fructose 1,6-bisphosphatase, FBPase1, which converts fructose 1,6-bisphosphate to fructose 6-phosphate. FBPase1 activity is regulated allosterically by various metabolites, including AMP, fructose 2,6-bisphosphate, and citrate. Furthermore, the phosphorylation state of FBPase1, mediated by protein kinase A, modulates its activity. Alongside FBPase1, other enzymes, such as pyruvate carboxylase and phosphoenol pyruvate carboxykinase, also play crucial roles in controlling gluconeogenesis. During gluconeogenesis, different substrates are utilized to generate glucose. Amino acids, obtained from protein breakdown, serve as significant precursors. Alanine, aspartate, and glutamate are the primary amino acids that contribute to gluconeogenesis. Furthermore, lactate, produced as a byproduct of anaerobic glycolysis, can also be converted to glucose. Glycerol, derived from triglycerides through lipolysis, is another essential substrate utilized in gluconeogenesis. Understanding the diverse array of substrates used in this process provides insights into the adaptability and flexibility of gluconeogenesis. The pentose phosphate pathway, an alternative route of glucose metabolism, intersects with gluconeogenesis. This pathway generates ribose 5-phosphate for nucleotide synthesis and produces NAD for reductive biosynthesis reactions. The enzyme glucose 6-phosphate dehydrogenase plays a pivotal role in governing the flow of carbon through the pentose phosphate pathway. By understanding the interconnectedness of the pentose phosphate pathway with gluconeogenesis, we can appreciate the complex interplay of metabolic pathways in maintaining cellular homeostasis. Anaplerotic reactions replenish the intermediates of metabolic pathways. These reactions are vital for sustaining gluconeogenesis and preventing depletion of essential metabolites. Reactions, such as the carboxylation of pyruvate to oxaloacetate by pyruvate carboxylase, fulfill this crucial role. This replenishment ensures the availability of substrates for gluconeogenesis and prevents the accumulation of intermediates that could disrupt the metabolic equilibrium. In addition to amino acids, lactate, and glycerol, certain molecules serve as critical gluconeogenic precursors. Propionate, a metabolite derived from odd-chain fatty acid oxidation, is one such example. Moreover, other compounds, including pyruvate, oxaloacetate, and alpha-ketoglutarate, can enter the gluconeogenic pathway via anaplerotic reactions. Understanding the contribution of these precursors enhances our comprehension of the intricacies involved in gluconeogenesis. The regulation of key enzymes in gluconeogenesis is tightly controlled to maintain glucose homeostasis. Hormonal regulation plays a crucial role, with glucagon stimulating gluconeogenesis through the activation of cyclic AMP-dependent protein kinase A. Conversely, insulin suppresses gluconeogenesis by inhibiting several key enzymes. Transcriptional regulation also influences gluconeogenesis, with transcription factors such as FOXO1 and CREB binding protein modulating gene expression. Unraveling the regulatory mechanisms allows us to gain insights into the fine-tuning of gluconeogenesis under different physiological conditions. Hormones exert significant influence over gluconeogenesis. Glucagon, released by the pancreas during fasting or low blood glucose, stimulates gluconeogenesis. It acts through the cyclic AMP-dependent protein kinase A pathway and results in enhanced gene expression and activation of key enzymes. On the other hand, insulin, secreted when blood glucose levels are elevated, inhibits gluconeogenesis by impeding the expression and activity of gluconeogenic enzymes. The dynamic interplay of these hormones ensures the maintenance of glucose levels within a narrow physiological range. Gluconeogenesis plays a crucial role in maintaining energy balance and control. During prolonged fasting or intense exercise, when glucose reserves become depleted, gluconeogenesis becomes paramount in replenishing glucose levels. Additionally, the energy cost of gluconeogenesis itself must be considered, as it requires ATP and consumes diverse substrates. Proper coordination of energy consumption and generation is essential to avoid metabolic imbalance and ensure the continuous supply of glucose to vital organs. Although the liver and kidneys are the primary sites of gluconeogenesis, this pathway is also present in other tissues, such as the small intestine and brain. In these tissues, gluconeogenesis serves specific physiological roles, including the maintenance of glucose homeostasis and provision of energy for cellular processes. Understanding tissue-specific regulation and functions of gluconeogenesis broadens our understanding of its implications beyond the traditional sites of its occurrence. Genetic disorders affecting the enzymes involved in gluconeogenesis can have severe consequences. Examples include deficiencies in pyruvate carboxylase, fructose 1,6-bisphosphatase, and glucose 6-phosphatase. These deficiencies disrupt gluconeogenesis and can lead to hypoglycemia, metabolic acidosis, and neurological complications. Studying these disorders enhances our understanding of the role each enzyme plays in this intricate metabolic pathway. Transcriptional regulation of gluconeogenesis involves complex networks of transcription factors and coactivators. Sterol regulatory element binding protein 1C and peroxisome proliferator activated receptor gamma coactivator 1-alpha are key regulators of hepatic gluconeogenesis. They coordinate the expression of gluconeogenic enzymes and contribute to the tissue-specific control of gluconeogenesis. Unraveling these transcriptional networks provides insights into the exquisite control of this enzymatic pathway. Gluconeogenesis has important clinical applications. It is relevant in diseases such as diabetes, where impaired regulation of gluconeogenesis contributes to hyperglycemia. Additionally, cancers often exhibit increased gluconeogenic flux to meet the high demand for energy and biomass. Understanding the aberrant regulation and utilization of gluconeogenesis in various pathological conditions opens avenues for developing targeted therapies and diagnostic approaches. In conclusion, gluconeogenesis is a remarkable metabolic pathway that ensures glucose availability and plays a crucial role in energy homeostasis. Its complex regulation, intricate interconnections with other metabolic pathways, and implications in various physiological and pathological conditions make it a fascinating subject of study. By comprehending the detailed mechanisms and functions of gluconeogenesis, we can unravel the complexities of glucose metabolism and its importance in human health and disease. Never cease to marvel at the wonders of biochemistry.